So we'll wrap up Chapter 9 by taking a look at the characteristics that are used to classify bacteria, and then talk a little bit about some of the rules for naming bacteria. So when we look at macroscopic organisms, a lot of the criteria that are used to classify them just don't work as classifying tools for bacteria. So here are some of the criteria, or the primary criteria, that are used to classify and to identify microbial life. The first is the morphology, so the size of the cell and the shape of it. The staining properties, so the characteristics primarily of the cell wall and perhaps the capsule that's around it. So gram staining, acid-fast staining, all of those differential stains that we had talked about in previous chapters. Their growth, so as they grow, do they develop pigments, sort of like Serratia marcescens, the one that we were working with in lab this week that makes that red pigment. The shape of the colony, how when they're in broths, how do they cluster? Do they come apart as they divide? Their nutritional requirements, so we try to grow them out and we discover, oh, we can't grow this bacteria on this medium. And so we try, say, a blood agar and we discover that, oh, it's a fastidious bacterium. Or do they ferment? Do they go through the processes of fermentation when they're grown out? Do they require sunlight in order to get their nutritional or energetic needs met? Their physiology, so what the critical temperature ranges are, their ability to tolerate osmotic stress, the pH that they can handle, what antibiotics they respond to, what phages they respond to, those sorts of things. So to classify them, though, so to continue with that list of criteria, sorry, I got a little distracted there. To continue with that list of criteria, we would add to it biochemistry, so the what the cell wall is composed of, the RNA and the proteins associated with the RNA, the inclusions that are found in the cells, the antigens that are found on the cell surface, and then molecular analysis, so looking at the DNA itself or the RNA itself, so the genetics of it, the GC ratios, so their GC percentages, the ability of the DNA to hybridize. Serology, so that's a term related to immunization, or immunology, not immunization, but immunology. So the use of antibodies to look for specific proteins or specific sequences of amino acids. The phage typing work that we can do. And then, looking at the RNA and the ribosome. So all of these categories and criteria, what we discover can then be used as either identification tools, if we already have a way of knowing the characteristics of organisms around all of these criteria, or as a way of organizing them, creating phylogenies, creating these evolutionarily based classification systems. So using these kinds of criteria, then, we can identify bacteria to their genus and their species. So remember, kingdom, phylum, class, order, family, genus, and then specific epithet, or the naming. So a species, then, is the collection of strains that share many common features and differ significantly from other strains. So the kind of two levels of criteria. First, any strain that, and we should probably identify what a strain is. Those are the descendants of a single cell that was found, that we isolated into pure culture. So if we have two strains that we're comparing to each other, if they share lots of common features and they differ from a lot of other organisms that we know about, then we would start calling those species. And the type, so within these ideas of strains, there are what are termed type strains. So if a biologist, a microbiologist, discovers a new species or a new species of microbe, then they would collect all that information, all the information about how they respond to all the criteria, the food they grow on, the pH tolerance, the characteristics of the DNA that can be used to differentiate it, or that were used and are used to differentiate it from other organisms. All that information is collected, and then a preserved specimen of that type, of that strain, is then, it's taken and preserved. So we had talked about how we preserve bacteria and then keep them in type collections. And so this, and for instance, we talked about the American type culture collection. So when we talk about a type, that is what we're talking about is a species, and the type strain is the strain that is used to identify a species. And so that type is then preserved forevermore, so that if we ever have questions about relatedness between the type and other strains that we have, then we can go directly to the source and do comparisons. So we take all of the, so these similarities that we see, let me back up for a second, the similarities that we see in the DNA and in the proteins have really proven to be reliable strategies for assigning individual strains to existing species, or if it's a new strain that we have never seen before, never been described before, then for establishing a new genus or of a new species. And it's really difficult, it turns out, to get much higher than genus. We talk about families of bacteria, but above families, there's not a lot of clustering that's done. And for all of those reasons, because of the challenge that we have in ascertaining what the evolutionary relationships are among bacteria, and all of that lateral transfer of information among bacteria that we talked about. So we generally don't see much classification above the family when we're looking at microbes. And who knows? I'm sure that taxonomists would love to be able to do that, to get that kind of, to go that deep in how we classify. And over time, I suspect we'll see the development of strategies, of techniques that might allow us to do that kind of classification above that level of family. Okay, so what are the tools then that microbiologists can use to identify organisms that they have discovered, for instance, or to place the organisms that they are working with within an evolutionary perspective, a phylogenetics perspective? And what is used is two sets of books. There's the Bergey's Manual of Determinative Bacteriology, and that one we have a copy of in our lab. So if you are interested in seeing it, I really encourage you to say something to me, and I'll drag it out. And I keep dragging it out, and every once in a while somebody will be like, "Oh yeah, show me what's in there." So be one of those people. Come and ask me to take a look inside Bergey's Manual. And then there's a second set of volumes called Bergey's Manual of Systematic Bacteriology. The latest edition of Bergey's Manual of Determinative Bacteriology is the 9th edition, so we own that one. There are 6 volumes in the Bergey's Manual of Systematic Bacteriology, so it's a much more voluminous bit of authorship. So in the Bergey's Manual of Determinative Bacteriology, that's the one that's used to help distinguish amongst species, to identify if you have an unknown species in front of you, then you would use Bergey's Manual in order to determine what you've got. And it's built on a classification scheme that divides the bacteria into 4 groups based on their cell wall structure: gram-positive, gram-negative, those that have no cell wall, they're referred to as mycoplasmas. And then those that have have a cell wall, but it is atypical. For instance, it might not have peptidoglycan in it. So there are three main sections then that then distinguish amongst these four different types of characteristics. And as we go to name bacteria, or to determine, if you hear somebody use a term and you're like, oh, what does that mean? I was saying family before. I meant order. Yeah, we do place things into orders. Class, order, order and family. So I apologize for that. Consider me correcting that mistake. So there are some internationally agreed upon rules for how, there's an international commission on the naming of bacteria. And but even as we learn more about the bacteria that we have in front of them, we will frequently change names of bacteria to get them more organized appropriately, or even move groups amongst these higher level taxas. So here are some of the rules. Order names always end with ales, A-L-E-S. So, for instance, the order Lactobacillales or the order Rickettsiales. Family names always end with this suffix ace. I know it sounds like I'm spelling something, but I'm really saying it. You say A and then C, that's the CE. And anytime you see A and E together, you would pronounce that as E as well. So A-C-E. ACE is how you would say that. So there's the family Listeria ACE and the family Clostridiae ACE. Clostridiae ACE, you would actually say it kind of together like that rather than the way I was doing it. So the Listeria bacteria, named for Lister, who you've heard of, and Clostridium, the Clostridium bacterias that are named, that cause things like botulism, or botulinum toxin, the Botox that we inject into our faces to look more youthful and vigorous, or the one that causes tetanus, Clostridium tetani. Those are family names, and they would end with ACE. So ALES, orders, ACE, families.